The testis is organized into a network of seminiferous tubules. Within these tubules, spermatogenesis occurs. Spermatogenesis is the production of mature haploid sperm cells through meiotic cell division. A spermatogonium deep in the epithelium of the seminiferous tubules gives rise to a primary spermatocyte. This cell contains 46 chromosomes. A primary spermatocyte undergoes the first meiotic division, producing two identical secondary spermatocytes. Each secondary spermatocyte contains 23 chromosomes. The secondary spermatocytes complete meiosis, giving rise to haploid spermatids. Each spermatid contains 23 chromosomes. The spermatids then develop into mature sperm. The penis is filled with vascular erectile tissues that allow it to exist in a flaccid or erect state. An erection is achieved through regulation of blood pressure within the penis. Upon psychic or tactile sexual stimulation, venous blood flow out of the penis decreases, while arterial flow into the erectile tissues increases. The size of the erectile tissues is limited by a dense covering called the tunica albuginea. Engorgement of these tissues with blood results in a stiffening of the penis. Circumcision is the removal of the foreskin or prepuce of the penis. A circumcision is generally performed for hygienic purposes because the glans penis is easier to clean if it is exposed. The first of two incisions is made vertically across the prepuce. The second incision is made with the help of special surgical tools. This incision is located around the base of the prepuce. Now the prepuce is removed, exposing the glans penis. Each month, the uterus undergoes a menstrual phase if pregnancy fails to occur. During the menstrual phase, the endometrium is shed along with bleeding that occurs due to the rupture of the endometrial blood vessels. The proliferative phase lasts for about 9 days. It is during this phase that the endometrium thickens. The secretory phase follows ovulation, and it is during this phase that the glands of the uterus make glycoproteins that serve as embryonic nutrition prior to implantation. The secretory phase is approximately 13 days long. Under the regulation of the pituitary hormones, luteinizing hormone, LH, and follicle-stimulating hormone, FSH, follicles develop within the ovary. Primordial, primary, and secondary follicles contain an oocyte that is suspended in prophase of the first meiotic division. Growth in these follicles is due to an increase in the number of granulosa cells. Secondary follicles develop an antrum that contains the secretions of the granulosa cells. Within the mature follicle, the first meiotic division is completed. A sharp increase in the LH level stimulates ovulation. After ovulation, the remaining granulosa cells and surrounding cells form a corpus luteum. If pregnancy occurs, the corpus luteum is maintained. If pregnancy does not occur, the corpus luteum slowly degenerates to form a corpus albicans. The ovary is the site of the development of the oocyte. Within the ovaries, follicles develop. Each follicle houses an oocyte, which matures simultaneously with the follicle. The primary follicle houses an oocyte encapsulated by a coating called the zona pellucida. It also contains a layer of granulosa cells that surrounds the oocyte. The secondary follicle contains many more granulosa cells and an accumulation of follicular fluid that is secreted by granulosa cells. This fluid coalesces in the follicle to form the antrum. A mature follicle is much larger than a secondary follicle, and the antrum occupies a greater portion of the follicle. It is within the mature follicle that the first meiotic division of the oocyte is completed, to produce a secondary oocyte and the first polar body. The secondary oocyte receives most of the cytoplasm from the cell division. However, the nuclei and the chromosomes are divided equally. The polar body degenerates. The mature follicle ruptures at ovulation to release the secondary oocyte and a corona of granulosa cells into the uterine tube. The second meiotic division cannot be completed unless the secondary oocyte unites with the sperm cell. As a result of this division, a second polar body is formed, and the oocyte and sperm nuclei combine to form a zygote. In the normal male reproductive tract, mature sperm are stored in the epididymides. They travel through the vas deferens into the urethra and exit the penis during ejaculation. Vasectomy is a surgical contraceptive procedure whereby the vas deferens are permanently divided, preventing the spermatozoa from leaving the epididymis. Following vasectomy, a man can only ejaculate seminal fluids that are free of sperm cells. In the normal female reproductive tract, the ovary releases ova into the uterine tube, the passageway which carries them to the uterus. The uterine tube is a common site of fertilization of the ovum. Tubal ligation is a surgical contraceptive procedure whereby the uterine tubes are permanently divided. This division creates a barrier between sperm and ova that prevents fertilization.